Hello and welcome to an exciting lecture on mutations. So this is where science kind of gets really fun because we start to talk about what can go wrong. So we've already learned about DNA replication and protein synthesis, and when something goes wrong and there's a mistake made in those two processes, we can get something called mutations. And a lot of people think of mutations as being bad, right? You get a disease or an undesirable characteristic, right? Two heads in the case of this snake. But in some cases, if the mistake leads to a protein that helps an organism survive, it can actually lead to evolution. So mutations are actually the driving force of evolution and all life on Earth. So it becomes really interesting. So let's take a closer look at how this happens. So mutation is a change in the DNA sequence that affects genetic information. So these mistakes usually occur during DNA replication, during S phase of the cell cycle when the whole DNA is getting replicated. The DNA mistake codes for a different or non-functioning protein in the phenotype, right? So the physical characteristics. So that protein, that normal functioning protein, maybe it's a protein that carries oxygen in blood cells. If there's a mutation in that protein, it's either not going to work as well or it's not going to work at all, depending on how severe the mutation is. And one important thing to note about mutations is that they're only passed down from one generation to another when they occur in the sex cells, so in the sperm or the egg cells. So for example, if I'm sitting out in the sun all summer and I get some mutations in my skin cells because I'm getting damage from the sun, those mutations might harm me and might harm my skin, but they're not going to be passed down to my offspring because they're not going to be in my egg cells. They're only in my skin cells. Okay? So now let's look at an example. So will this mutation, we have a normal DNA sequence here. A mistake was made here during DNA replication, right? Where there was supposed to be a C, now there's a G. And I want to know, so I want you to sequence this out, transcribe and translate it into an amino acid sequence, and see if we have a change in the protein. Okay, so if you need to Google amino acid wheel, you can to help you here. Give this a shot. All right, so hopefully you were able to transcribe the DNA into RNA, in which you should have gotten this code here, right? A pairs with U on RNA, T pairs with A, G pairs with C, so on and so forth. And then when you use the amino acid wheel, right, if you use UAC, I'm going to go here. We start with a U, so you're in the U section of the wheel. UAC, we get the amino acid tyrosine. All right, so you should have, the first one should be tyrosine, glutamine, and threonine. But when the mistake was made here, instead of a glutamine, what should have been a glutamine is now a histidine. Okay, so now we have two different amino acid sequences. So you can imagine when these proteins are put together in the ribosome and the endoplasmic reticulum, they're not going to function the same, right? Because they have a different code. So they are no longer the same protein. Okay, so this is how we get a mutation. So one example would be sickle cell anemia, you've probably heard of. So this is a normal red blood cell. It's supposed to be nice and round and look like a pretty little red donut. And what this cell does is it has a really good job at holding oxygen and transporting oxygen around our body. But in someone who has sickle cell anemia, the proteins that make the hemoglobin and that make this cell are not correct. So instead of being a nice round plump juicy cell here that is really good at carrying oxygen, it's this sickle shape and it is not as efficient at carrying oxygen. So it still functions enough that the person is alive, but not well enough that the person is completely healthy. They're going to have some health problems. Here's another example. A lot of people are colorblind, right? If you can't see this number 29 in the middle here, you are colorblind. And a lot of people are. And that is caused by a mutation in our eyes. Another example is people who are lactose intolerant. Actually, being able to digest milk protein is actually the mutation. So if you are not lactose intolerant, you're the one with the mutation that's actually beneficial for you because you can eat dairy and cheese and all the delicious dairy things that you like. Pretty interesting. All right, so let's start to take a look at the types of mutations. There's a few different types. The first type, very simple, and it's called a point substitution mutation or a point mutation. So this is a gene mutation that involves changes in one or just a few nucleotides. So if we look at this example here, we have the normal DNA, the original DNA. When the complementary DNA strand was made, there was a mutation where there should have been a T, here there's a G. So when it was transcribed into RNA, we have one point that has been changed, right? So this is a point substitution. Now there's a couple different types of point mutations. Let's look at them. First one is called a missense mutation. So this is a point mutation that results in a change in the amino acid sequence. Okay, so when we, just like the example we just did a second ago, that single point change from a C, what should have been a C is now, or stays a C, it should be a G, and here it's a C. That led to one amino acid change in the DNA sequence. Okay, so it should be this, but instead it's this. So that is a missense mutation, leads to a change in the amino acid sequence. Second type of point mutation, again, we're still a point mutation, so we still have a 1 point change in the DNA sequence, but this time it results in an early stop codon. Remember on the amino acid wheel, some of the amino acids aren't actually amino acids, they're stops. So some of the codons code for a stop codon, which means the protein is done, the ribosome stops working on it, folds it up, sends it off on its way. So if we have an early stop codon, the amino acid sequence is going to be shorter than it's supposed to be. So this usually ends with a non-working protein because it's missing a lot of amino acids that it should have. Okay, so the stop comes earlier than it should. And the last type of point mutation is called a silent point mutation. So this is a mutation that does not result in a change in the amino acid sequence, right? It's silent, doesn't make any noise. So even though there's a change in the bases, that change, GAC and GAU, still code for the same amino acid, right? So if we look here, GAC and GAU both code for aspartic acid. Okay, so there's no change. So that protein, we will never know that that person had a mutation in their genes because the protein is still working exactly the same. Okay. Next type of mutation is called a frameshift mutation. So this is a mutation that shifts the reading frame or the codons of the genetic message by inserting or deleting a nucleotide. So these are easy to identify because you either are going to have an extra base at the end, or you're going to have one less, right? Because we're either adding an extra one, inserting, or deleting. So you can say you can see that we have 1, 2, 3, 4 codons here. But in the mutated one, we've got 1, 2, 3, 4 codons, and then we have an extra one over here, an extra base. And by this G being inserted, now all of the codons that come after the mutation are going to be different, right? So this should be AGU, GUG, UGA, and look, they're completely different. Okay, so this is going to lead to major changes in the amino acid sequence. Okay, so two basic types, again, point mutation and frameshift mutation. And if we're looking at point mutations, there's three types of point mutations: missense, nonsense, and silent. All right, now let's talk about what causes mutations. So some mutations just happen randomly. Unfortunately, they do. Other mutations can be caused by environmental factors, so things like radiation, right? Sitting in the sun too long, going in tanning beds. That UV radiation can cause mutations in our DNA, which can cause our cells to not function properly in our skin and can lead to things like skin cancer. X-rays, right? Being exposed to too many X-rays or radiation, MRIs, CT scans, things like that can cause mutations in the DNA. Certain chemicals, so smoke, certain drugs, some food preservatives, unfortunately. By barbecuing your food and burning it, that char can cause mutations. And then some infectious agents as well. So there's some viruses and bacteria that can actually cause mutations as well. Okay, I know a lot of young people now get the Hpv vaccine when they're young. That's because HPV is a virus that has been known to cause cancer because it can cause mutations in the DNA. Okay, so here's just a little diagram of that.